Bismillahirrahmanirrahim. Assalamu alaikum. My name is Alishba Zainab from BS Chemistry. Today my presentation topic is organoid intelligence. So these are the presentation outlines here. First one, introduction, background concepts, scientific foundations, technological integration, applications of organoid intelligence, case studies, ethical and philosophical considerations, challenges and limitations, future direction, conclusion, references. So let's start this. Introduction to organoid intelligence. What is organoid intelligence? Organoid intelligence refers to the use of brain organoids, three-dimensional cluster of human neurons grown from stem cells as biological substrates for learning, adaptation, and computation. These organoids mimic aspects of the human brain, including layered structures, synaptic connectivity, and spontaneous electrical activity, unlike traditional artificial intelligence, which relies on silicon-based processors and algorithmic logic. OI leverages the natural properties of living neurons to process information. This approach is part of a broader movement toward biocomputing, where biological systems are used to perform tasks typically handled by digital machines. Key characteristics of OI: biological substrate uses living neural tissues instead of silicon chips. Adaptive learning: neurons can rewire and strengthen connections based on experience. Energy efficiency: biological systems consume far less energy than digital processors. Human-like cognition: organoids may offer insights into memory, decision making, and consciousness. OI is not about creating full brains in a dish, rather it's about using simplified neural systems to explore how biological intelligence works and how it can be harnessed for practical applications. Why is organoid intelligence important now? The urgency and relevance of OI stem from several converging factors in science, technology, and society. Limitations of traditional AI: silicon-based AI systems are reaching physical and energy limits. Deep learning models require massive dataset and computational power. AI lack the organic adaptability and contextual awareness of biological systems. OI offers a new path forward, one that mimics the brain efficiency and flexibility. Advances in stem cell technology: the discovery of induced pluripotent stem cells by 2006 revolutionized regenerative medicine. iPSCs allow researchers to create patient-specific organoids ethically and efficiently. Organoids can now be grown to mimic various brain regions and functions. These breakthroughs make it possible to study human-like neural systems in vitro. Emerging brain-machine interfaces. Technologies like multi-electrode arrays enable researchers to stimulate and record from organoids. Feedback loops allow organoids to learn from experience, similar to reinforcement learning. Hybrid systems combining organoids and AI are being developed for tasks like game playing and drug testing. This integration of biology and electronics is opening new frontier in computing, medical and ethical imperatives. Organoids are being used to model diseases like Alzheimer, autism, and Parkinson. They offer personalized platforms for drug testing and treatment development. As organoids become more complex, ethical questions about sentience and moral status arise. OI is not just a technological innovation, it's a tool for understanding and improving human health. Background concepts. Definition of brain organoids. Brain organoids are three-dimensional, miniature, simplified versions of the human brain grown into vitro from stem cells. They are not full brains but rather clusters of neurons and supporting cells that self-organize into structures resembling parts of the brain, such as the cerebral cortex, hippocampus, or retina. Key features: derived from human stem cells, often induced pluripotent stem cells, self-organizing. Cells arrange themselves into layered structures similar to those found in the brain. Functional activity. Organoids exhibit spontaneous electrical activity from synapses and can respond to stimuli. Used for research, neuroscience, disease modeling, drug testing, and nerve biocomputing. Brain organoids provide a unique platform for studying brain development, neurological disorders, and the biological basis of cognition in a controlled lab environment. How organoids are created? Created brain organoids involve a multi-step process that transforms stem cells into functional neural tissues. First one, stem cell reprogramming. Scientists start with adult cells, skin or blood cells and reprogram them into induced pluripotent stem cells using transcription factors like so on. iPSCs behave like embryonic stem cells and can differentiate into any cell type. Neural induction iPSCs are exposed to specific signaling molecules that guide them to become neural progenitor cells precursors to neurons and glial cells. This step mimics early brain development in the embryo. 3D culture formation. Neural progenitors are embedded in a gel-like matrix and placed in a spinning bioreactor. The bioreactor provides nutrients and oxygen while promoting self-organization into 3D structures. Maturation. Over several weeks, the organoid develop neuronal layers, synaptic connections, and spontaneous electrical activity. Depending on the culture conditions, organoids may form specialized regions like the cerebral cortex or retina. These processes allow researchers to grow brain-like tissue that can be used to study development, disease, and even perform computational tasks in the emerging field of organoid intelligence. Key differences. Biological versus digital. OI uses living neurons that adopt and learn through natural processes while traditional AI relies on mathematical models and digital logic. Energy use. Biological systems are far more energy efficient than silicon chips, which require significant power for training and inference learning. OI mimics human learning through synaptic changes, whereas AI uses structured data and optimization algorithms. Ethics. OI raises new ethical questions about the use of human-derived tissue, potential sentience, and moral status. Organoid intelligence could relate to computing systems that are more human-like in their learning and adaptability, though they are currently limited in scale and complexity compared to digital AI. Scientific foundations of organoid intelligence. Neuroscience behind organoids. Brain organoids are derived from human stem cells and cultured to mimic early brain development. These 3D structures contain neurons and glial cells arranged in layers similar to the cerebral cortex. Neuroscientifically, they offer a simplified but biologically relevant model of the human brain. Organoids exhibit spontaneous electrical activity. Neurons fire without external stimulation, indicating intrinsic network behavior. Synaptic connectivity. Neurons form synapses, allowing communication and signal propagation. Neurodevelopmental patterns. Organoids show gene expression and structural organization akin and to fetal brain development. These features make organoids valuable for studying neurogenesis, disease modeling, and most recently biocomputing. Unlike traditional neural culture, organoids provide a 3D environment that supports more complex interactions and maturation. Synaptic activity and learning in vitro. Synaptic activity is the foundation of learning and memory in biological systems. In vitro organoids demonstrate synaptic plasticity, the ability of synapses to strengthen or weaken over time based on activity. This is the biological basis for learning. Long-term potentiation. A sustained increase in synaptic strength following repeated stimulation observed in organoid models. Neural oscillations, coordinated rhythmic activity across neurons similar to brain waves in vivo. These behaviors suggest that organoids can encode information and adapt to stimuli. Researchers use multi-electrode arrays to stimulate organoids and record their responses. Over time, organoids modify their firing patterns, indicating a form of learning. In one study, organoids exposed to repeated electrical stimuli showed enhanced responsiveness, mimicking associative learning. This opens the door to using organoids as biological processors capable of adapting to tasks. Key experiment: organoids playing Pong. One of the most groundbreaking demonstrations of organoid intelligence was conducted by Cortical Labs in 2022. Researchers trained a cluster of human neurons grown in a dish and a dish brain to play the classic video game Pong. Experiment setup: electrode interface. The organoid was placed on a grid of electrodes that was stimulated and recorded neural activity. Game environment: the organoid did not see the game visually, instead it received electrical signals representing the ball position and velocity. Feedback loop: when the organoid responded correctly, it received positive feedback. Incorrect responses led to negative feedback. Result: within minutes, the organoid began adjusting responses to improve performance. It demonstrated goal-directed behavior, modifying its activity based on outcomes. The organoid performance was significantly better than random chance, indicating learning. Dr. Brett Kagan, Chief Scientific Officer at Cortical Lab, stated, "We have shown we can interact with living biological neurons in such a way that compels them to modify their activity, leading to something that resembles intelligence." Scientific implications. Proof of concept: biological neurons can be trained to perform computational tasks. Adaptive learning: organoid change its behavior based on feedback, a hallmark of intelligent system by computing potential. This experiment suggests that organoid could be used in future computing systems that learn and adapt organically. Ethical considerations: the experiment sparked discussion about the moral status of organoid. While each brain lacked consciousness, its ability to learn raised questions about future development. Broader impact on neuroscience and AI. The Pong experiment related studies have profound implications. Neuroscience: organoid offer a controllable model to study learning, memory, and neural dynamic. AI research: biological systems may inspire new algorithms or hybrid models combining digital and organic intelligence. Medical application: understanding how organoid learn could improve treatment for neurological disorders. Organoid intelligence bridges the gap between biology and technology, offering a new paradigm for understanding and harnessing intelligence. Technological integration in organoid intelligence. Organoid intelligence represents a radical shift in how we think about computing. Instead of relying solely on silicon-based processors and digital algorithms, OI uses living brain organoid clusters of human neurons grown in vitro as biological substrates for learning, adaptation, and computation. To harness their potential, scientists must integrate these organoids with electronic systems that can stimulate, monitor, and interpret their activity. This section explores how that integration works and what it means for the future of hybrid intelligence. Interfacing organoid with electronics. To interact with brain organoid, researchers use specialized tools that allow them to send signal to the organoid and record its responses. The most common interface: the multi-electrode array, a grid of microscopic electrodes embedded in a dish where the organoid is cultured. Multi-electrode arrays. Multi-electrode arrays contain dozens to hundreds of electrodes that can simultaneously stimulate and record from different regions of the organoid. They detect extracellular electrical signals such as action potentials and local field potentials. Meta-electrode arrays allow researchers to map neural activity, observe patterns, and identify regions of interest. Stimulation technique. Electrical stimulation. Electrodes deliver pulses to specific regions of the organoid to mimic sensory input or trigger learning. Optogenetics. In some cases, neurons are genetically modified to respond to light. allowing precise control using laser or LEDs. Chemical stimulation, neurotransmitter or drug can be applied to modulate activity and study responses, recording and feedback. Meta-electrode array record the organoid electrode electrical responses in real time. These signals are analyzed using software tools to detect spike oscillation and network dynamics. Feedback loop can be created where the organoid performance influences future stimuli essential for learning tasks. This interface transforms the organoid from passive biological sample into an active participant in computational experiments. Hybrid system, biological and digital intelligence. One of the most exciting aspects of organoid intelligence is the potential to create hybrid systems that combine the adaptability of biological neurons with the precision and scalability of digital AI. What is a hybrid system? A hybrid system in OIs consists of a biological component, the brain organoid capable of learning and adapting through synaptic plasticity, a digital component, software algorithms, machine learning models, and control systems that interpret organoid activity and adjust stimuli. These systems work together in a feedback loop. The digital system sends input to the organoid processes the input and generates a response. The digital system analyzes the response and determines the next input. Benefits of hybrid intelligence adaptability. Biological neurons can rewire themselves in response to experience, offering dynamic learning. Energy efficiency. Neurons consume for less energy than silicon chip, making biological computing more sustainable. Human-like recognition. Organoids may process information in ways that resemble human thought, offering insight into consciousness and decision making. Example, dish brain playing Pong. In 2022, researchers at Cortical Lab created a hybrid system where a brain organoid was trained to play the video game Pong. The digital system translated the game environment into electrical signals which were sent to the organoid. The organoid responded by adjusting its firing pattern to control the paddle. Over time, it learned to improve its performance, demonstrating goal-directed behavior. This experiment showed that hybrid system can perform tasks traditionally reserved for digital AI, but with biological adaptability. Data input-output mechanisms. To make organoids useful for computation, researchers must establish reliable method for sending data into the organoid and extracting meaningful output. This involves translating digital information into biological stimuli and vice versa. Data input. Input to the organoid can take several forms: electrical pulses delivered through meta-electrode array to stimulate sensory input or trigger learning. Chemical signal, neurotransmitter or drugs can modulate activity and mimic biological conditions. Environmental cues, temperature, light, mechanical stimulation may influence behavior. The input must be encoded in a way that the organoid can interpret. For example, in the Hong experiment, the position and velocity of the ball were translated into patterns of electrical stimulation. Data output. Output from the organoid are typically recorded as fine-grained sequence of action potential from the individual neurons, oscillatory pattern, rhythmic activity across neural networks, network dynamic changes in connectivity, and synchronization between regions. These outputs are analyzed using computational tools to identify patterns, measure learning, and assess performance. Machine learning algorithms may be used to classify responses or predict future behavior. Feedback loops. A key feature of OI system is the closed loop architecture where the output of the organoid influences future input. This allows the system to learn from experience and adapt over time. For example, if the organoid responds correctly to a stimulus, it receives positive reinforcement, a reward signal. If the response is incorrect, the system adjusts the input or provide negative feedback. Over time, the organoid modifies its activities to improve performance. This loop mimics biological learning and is essential for tasks like game playing, pattern recognition, or decision making. Challenges and innovations. While the integration of organoid with electronics is promising, it comes with challenges. Signal noise, biological signals are complex and noisy, requiring advanced filtering and analysis. Variability, each organoid is unique, making standardization difficult. Longevity, organoids degrade over time, limiting long-term experiment. Ethical concerns as organoids become more complex questions about sentience and moral status arrive. To address these issues, researchers are developing advanced meta-electroarrays with high resolution and sensitivity, AI-driven analysis tools to interpret neural data, standardized protocols for organoid growth and interfacing, ethical framework to guide responsible experimentation. Future directions. Technological integration in OI is still in its infancy, but future development may include wireless interfaces, allowing organoids to interact with external devices without physical connections, multi-organoid system connecting multiple organoids to stimulate brain regions or perform complex tasks, real-time adaptive system where organoids and AI co-evolve to solve problems collaboratively. These innovations could lead to new forms of computing, personalized medicine, and deeper understanding of the human mind. Applications of organoid intelligence. Organoid intelligence is a rapidly emerging field that leverages brain organoid lab-grown clusters of human neurons to perform tasks traditionally handled by digital computers. These organoids mimic a set of human brain structure and function, making them powerful tools for computation, biomedical research, and ethical exploration. Below are four major application areas where AI is poised to make a significant impact. Biocomputing and low energy computation. Traditional computing systems rely on silicon-based processors that consume vast amounts of energy, especially in artificial intelligence applications like deep learning. In contrast, biological neurons are incredibly energy efficient. A human brain with billions of neurons operate on roughly 20 watts, less than a light bulb. Organoid intelligence offers a new paradigm for biocomputing. Energy efficiency. Brain organoid can process information using minimal energy, making them ideal for sustainable computing. Parallel processing. Neurons naturally operate in parallel, enabling complex computations without the need for massive hardware. Adaptive learning. Unlike rigid algorithms, organoids can rewire themselves in response to stimuli, offering dynamic learning capabilities. In experiments like the pong playing dish, brain organoid demonstrated the ability to learn and adapt using feedback loops. This suggests that future biocomputing systems could use organoid to perform tasks such as pattern recognition, decision making, and even creative problem solving, all with the fraction of the energy required by conventional AI. Researcher envisions hybrid system where organoids handle adaptive tasks while digital components manage speed and scalability. These systems could revolutionize fields like robotics, astronautics, navigation, and real-time data analysis, disease modeling, and drug testing. One of the most promising applications of brain organoid is in personalized medicine because organoids can be derived from a patient's own cells. They offer a unique platform for modeling disease and testing treatment tailored to individual genetic profiles. Disease modeling. Organoids have been used to replicate conditions such as Alzheimer's disease. Organoids with Alzheimer-related mutations show plaque formation and neural neuronal death, helping researchers study disease progression. Parkinson or disease Dopaminergic neuron loss can be modeled in organoid to explore therapeutic interventions. Autism spectrum disorder organoid derived from patients with autism reveal differences in neural connectivity and development. This model allows scientists to observe how disease affect brain structure and function real time without ethical and practical limitation of human or animal testing. Drug testing are designed to provide a controlled environment for testing pharmaceuticals safety and efficacy. Drug can be applied directly to organoid to assess their impact on neural activity and viability. High throughput screening. Multiple organoids can be tested simultaneously, accelerating drug discovery. Reduced animal testing. Organoids offer an ethical alternative to animal models especially for neurological conditions. By integrating organoid with AI system, researchers can automate data collection and analysis, identifying promising compounds faster and more accurately. This could lead to breakthroughs in treating complex brain disorder and tailoring therapies to individual patients. Brain development and neurodegenerative research. Brain organoids mimic earlier stages of human brain development, making them invaluable for studying how the brain forms, matures, and deteriorates. This has profound implications for both developmental neuroscience and aging research. Studying brain development organoids allow researchers to track neurogenesis, observe how stem cells differentiate into neurons and glial cells, map cortical layering, understand how brain regions organize and connect, investigate genetic disorder model conditions like microcephaly or lysencephaly by introducing specific mutations. These insights help scientists understand the origins of cognitive function and developmental disorder, potentially leading to early interventions and improved outcomes. Neurodegenerative research. As organoids mature, they can be used to study age-related changes, protein aggregation, monitor buildup of toxic protein like amyloid beta or synaptic loss, track the generation of neural connections over time. Inflammatory responses explore how glial cells contribute to neurodegeneration. This research is crucial for diseases like Alzheimer's, Parkinson, and Huntington, where early detection and targeted treatment could dramatically improve quality of life. Organoid also enable longitudinal studies where the same tissues observed over weeks or months, providing a dynamic view of brain aging and degeneration. Ethical AI and human-like cognition. As organoids become more complex and capable of learning, they raise profound ethical questions about the nature of intelligence, consciousness, and moral responsibility. Ethical AI. Organoid intelligence challenges traditional notion of artificial intelligence. Biological substrate. Unlike digital AI, OI uses living tissues, raising concerns about sentience and rights. Learning and memory. Organoid can adopt and retain information, suggesting a form of recognition. Moral status. If organoid exhibit goal-directed behavior or self-awareness, should they be protected? These questions demand interdisciplinary collaboration among scientists, ethicists, and policymakers. Guidelines must be developed to ensure responsible experimentation, informed consent for stem cell donors, and safeguard against misuse. Human-like cognition (OI) offers a window into how the human brain learns and thinks. Modeling consciousness or not may help identify the neural correlate of brain. Understanding decision-making, studying how organoids respond to stimuli can reveal mechanisms of choice and reasoning. Exploring creativity. Future experiments may test whether organoids can generate novel solutions or ideas. These insights could inform the development of ethical AI systems that mimic human cognition without replicating its vulnerabilities. They also raise philosophical questions about what it means to be intelligent, consciousness, or alive. broader implications. The applications of organoid intelligence or extend beyond science and medicine education. OI could be used to teach neuroscience and ethics in immersive, hands-on ways. Public policy governments may need to regulate OI research and its application. Industry by computing could transform sectors like finance, logistics, and cybersecurity. As OI matures, it will reshape how we interact with technology, understand the brain, and define intelligence itself. Case studies in organoid intelligence. Organoid intelligence is no longer just theoretical, it's being actively explored through groundbreaking experiments and platforms. These case studies highlight how researchers are training organoids to play games, using them to model neurodegenerative diseases and building scalable systems to harness their computational power. For experiment training organoids to play. One of the most iconic demonstrations of OI was conducted by Cortical Labs in 2022. Researchers created a system called DishBrain, which used human brain cells grown in a dish to play the video game. How it worked. The organoid was placed in a multi-electrode array that allowed researchers to send electrical signals and record neural responses. The game environment was translated into electrical stimuli. The organoid responded by adjusting its firing patterns to control the pattern. A feedback loop rewarded correct responses and penalized incorrect ones. Results. Within minutes, the organoid began modifying its behavior to improve performance. They demonstrated goal-directing learning, adapting to stimuli based on outcomes. The organoid performance exceeded random chance, indicating a form of intelligence. Dr. Brett Kagan, chief scientific officer at Cortical Labs, stated significances. This experiment proved that organoids can learn and adapt in real time. It laid the foundation for using organoids in biocomputing and interactive systems. It raised ethical questions about the nature of intelligence and the moral status of learning biological systems. Organoids used in Alzheimer's research. Brain organoids are also being used to model Alzheimer's disease, one of the most devastating neurodegenerative conditions. Because organoids can be derived from patient-specific stem cells, they offer a personalized platform for studying disease progression and testing treatment. Key applications: modeling play formation organoid with Alzheimer-related mutations show accumulation of myeloid beta plaque, a hallmark of the disease. Tracking neuronal death. Neuronal death. Researchers observe how neurons degenerate over time, mimicking the cognitive decline seen in patients. Testing drug potential treatment can be applied directly to organoids to assess their impact on neural health and activity. Advantages of human relevance: organoids reflect human biology more accurately than animal models. Ethical alternative: they reduce the need for invasive testing on humans or animals. Personalized medicine: organoids can be tailored to individual genetic profiles, improving treatment precision. These models are helping scientists understand the mechanism behind Alzheimer's and identify new therapeutic targets. They also allow for longitudinal studies where the same organoid is observed over time to track disease progression. Emerging platforms for OI experimentation: as interest in organoid intelligence grows, new platforms are being developed to support experimentation, scalability, and integration with digital systems. Cortical Lab pioneers of the dish brain system focused on building biological computing chips using organoid, exploring applications in gaming, robotics, and adaptive AI. Changer Project, a European initiative aimed at creating ethical and legal framework for OI support interdisciplinary collaboration across neuroscience, bioethics, and law, promote responsible innovation and public engagement. Neuro Neuronorm AI, academic and commercial platform for integrating organoid with AI system officer tool for data analysis, neural interfacing, and hybrid intelligence focused on applications in medicine, education, and cognitive research. APS Open Research published studies on training organoids with AI assistance investigate how organoids store information and adapt to the environmental changes. Both open access research and global collaboration platform feature standardized protocols, ensure reproducibility and consistency across labs, real-time monitoring use meta-electrode array and imaging tool to track neural activity. AI integration combine organoid responses with machine learning for enhanced performance. Ethical oversight include guidelines for consent usage and moral consideration. These platforms are essential for scaling AI research and translating it into practical application. They also foster collaboration between scientists, engineers, ethicists, and policymaker. Broader impact of these case studies. Together, these case studies demonstrate the versatility and promise of organoid intelligence. Scientific discovery, understanding how neurons learn and adapt outside the body, medical innovation, modeling diseases and testing experiments in personalized ways, technological advancement, building hybrid systems that combine biology and AI, ethical exploration, navigating the moral implications of intelligent biological systems. As OI continues to evolve, these examples will serve as benchmark for future research and development. Ethical and philosophical consideration in organoid intelligence. Organoid intelligence, is a frontier field that merges neuroscience, stem cell biology, artificial intelligence. It involves using brain organoid, tiny lab-grown clusters of human neuron as biological substrate for learning, adaptation, computation. As these organoids become more complex and capable of performing tasks, they raise profound ethical and philosophical questions. Could they become scientists? What right and responsibility do researchers have? How should society regulate and respond to this emerging technology? Could organoids become scientists? Scientists refer to the capacity to experience sensation, feeling, or consciousness. While current brain organoids are far from sentient, their increasing complexity and ability to learn raise important questions about future development. Current capability. Brain organoids today lack the structural complexity for full brain. They do not have sensory organ, blood supply, or the full architecture required for consciousness. However, they do exhibit spontaneous electrical activities and epiphysicitic goal-directed behavior. These features suggest a rudimentary form of information processing but not awareness, subjective experience. Future possibility. As organoids grow larger and more interconnected, they may begin to resemble more complete nervous system. If organoids begin to show signs of self-awareness, memory retention, or emotional responses, the question of sentience become unavoidable. Philosophical implications. What constitutes consciousness? Is it a threshold of complexity or a specific pattern of activity? For organoids become sentient? Do they deserve moral consideration? Could we unintentionally create suffering in others? These questions challenges our understanding of intelligence, ethical boundaries in science. Advances in Advances in vascularization, sensory interfacing, and long-term maturation could push organoids closer to functional brain-like states. Right and responsibilities in OI research. As with any research involving human-derived tissue, I demand a careful balance between scientific progress and ethical responsibility. Rights of organoids are typically derived from induced pluripotent stem cells which come from adult donor. Donor must give informed consent, understanding how their cell will be used. Ethical guidelines must ensure privacy, autonomy, transparency, responsibilities of researcher. Researcher must avoid causing harm even unintentionally. They should monitor organoid behavior for signs of complexity that might warrant ethical review. Experiments should be designed with minimal risk and maximum oversight. Moral status of organoids. If organoids begin to exhibit advanced cognitive function, their moral status may need to be reconsidered. Should they be treated like animal research with protection against suffering? Could they be considered a new form of life? These questions require informed ethicists, neuroscientists, legal experts, and the public. Institutional oversight. Research institutions must establish ethics committee to review all projects. The guidelines should evolve as the technology advances, ensuring responsible innovation. Collaboration across disciplines is essential to anticipate and address emerging concerns, regulatory and societal implications. OI is not just a scientific issue, it's a societal one. As the technology matures, government, industries, and communities must grapple with its implications. Regulatory framework. Currently, most countries regulate themselves, research, and AI separately. OI falls between these domains. New regulations may be needed to address creation and use of complex organized data, privacy, and ownership, especially with patient-derived model ethical boundaries for experimentation. International collaboration. OI research is global, requiring harmonized standards across border organizations like the Changer Project in Europe are working to create legal ethical framework for OI. International bodies WHO, UNESCO may play a role in setting norms and guidelines. Public engagement, public understanding of why is limited and misconceptions could lead to fear or resistance. Transparent communication is essential to build trust and support. Educational initiatives should explain what organized are, how they are used, what safeguards are in place. Societal impact. Hawaii could revolutionize medicine, computing, and education. It may also challenge our definition of intelligent life and identity as AI systems become more integrated into society. Questions will arise: should organoids be used in decision-making system? Could they be employed in surveillance, warfare, or other controversial domains? How do we ensure equitable access and prevent misuse? Ethical frameworks in practice. Several models can guide ethical decision making in OI. Precautionary principle. Avoid actions that could cause harm, especially when outcomes are uncertain in OI. This means monitoring organoid complexity and halting experiment if signs of a sentience emerge. Utilitarian ethics maximize benefit while minimizing harm. I offer immense potential in medicine and computing, but risk must be weighed carefully. Deontological ethics respect right and duty regardless of outcome. Donor consent, organized dignity, and research integrity are paramount. Virtue ethics focus on the character and intention of researchers, promote humility, responsibility, and compassion in scientific practice. These frameworks help navigate the moral landscape of AI and ensure that progress aligns with human values. Looking ahead, as organoid intelligence evolves, ethical and philosophical consideration will become increasingly urgent. Key priorities include monitoring the complexity, watch for signs of advanced cognition or sentience, updating guidelines, revise ethical standards as technology advances in engaging society, foster public dialogue and education, building safeguards, prevent misuse, and ensure equitable access. AI is not just about building a smarter system, it's about understanding what it means to be intelligent, conscious, and human. By addressing this question, now we can shape a future where science serves humanity with wisdom and care. Challenges and limitations of organoid intelligence. Organoid intelligence is a groundbreaking tool that merges neuroscience, stem cell biology, and artificial intelligence to create biocomputing systems using brain organoids. Miniature lab-grown neural tissues offer exciting possibilities from adaptive learning to disease modeling, but they also present significant challenges as researchers push the boundaries of what organoids can do. They must grapple with technical limitation, interpretive complexity, and ethical dilemmas. This section explored the key hurdles facing AI today. Scalability and reproducibility. Scalability challenges. One of the most pressing limitations in AI is the difficulty of scaling of brain organoid to perform more complex tasks. Current organoids are small, typically a few millimeters in diameter, and contain hundreds of thousands to a few million neurons. In contrast, the human brain has over 86 billion neurons and integrate regional specialization. Key issues include size constraints. Organoids lack blood vessels which limit their growth and nutrient delivery. Without vascularization, they cannot reach the size or complexity of a full brain connectivity. Larger organoids require more sophisticated neural network and interregional communication, which is difficult to replicate in vitro. Longevity organoids degrade over time, limiting long-term experiment and learning capacity. Efforts to overcome these challenges include bioengineering, vascularized organoids using synthetic scaffolds, or co-culturing with endothelial cells, connecting multiple organoids to stimulate brain region. Cortex-hippocampal interactions using bioreactor to enhance nutrient flow and oxygenation. Reproducibility issues. Scientific reproducibility is essential for validating results and building reliable systems. However, organoids are inherently variable due to biological and procedural factors. Sources of variability. Genetic differences. Organoids derived from different donors may behave differently. Culture conditions, small changes in temperature, medium composition, or timing can affect development. Batch to batch inconsistency. Even organoids from the same protocol can show divergent growth and activity. This variability makes it difficult to replicate environment compare results across labs or standardized OI platform. Researchers are working to develop standardized protocols for organoid generation and maintenance, quality control metrics to assess organoid maturity and functionality, automated system for organoid production monitoring. Until this solution are widely adopted, scalability and reproducibility will remain major barriers to OI broader application. Data interpretation and control. Brain organoids generate complex, noisy data that is difficult to interpret. Unlike digital systems which produce structured output, organoids communicate through electrical signals, synaptic activity, and network dynamics that require sophisticated analysis. Interpretation challenges. Signal complexity. Neural activity includes by spikes, oscillation, and bursts that vary across time and phase. Lack of benchmark. There are no universal standards for what constitutes learning or intelligence in organoid. Context dependence. Responses may vary based on prior stimulation, environmental conditions, or internal state. To address these issues, researchers use multi-electrode arrays to record activity across multiple regions, machine learning algorithms to detect patterns and classify responses, computational models to stimulate neural dynamics and predict behavior. Despite these tools, interpreting organoid data remains a challenge. It requires interdisciplinary expertise in neuroscience, data science, and bioengineering. Control limitation. Controlling organoid behavior is equally difficult. Unlike digital system which follows program instruction, organoid respond organ, organ, organically and unpredictably. Control challenges include stimulus response variability. The same input may produce different output depending on the organized state. Feedback loop. Creating adaptive system requires real-time monitoring and adjustment, which is technically demanding. Limited precision. Electrical stimulation affect multiple neurons, making targeted control difficult. Researchers are exploring optogenetic, using light to control genetically modified neurons with high precision closed-loop system where organoid output influences future input, enabling learning and adaptation. Hybrid interfaces combining biological and digital components to enhance control and responsiveness. Until this method refine data interpretation and control will limit the complexity and reliability of OI system. Ethical boundaries and public perception. As OI advances, it raises profound ethical questions and public concern. These issues must be addressed to ensure responsible innovation and societal acceptance. Ethical boundaries are not derived from human cells and can exhibit learning behavior as they become more complex. Questions arise about their moral status and treatment. Key concerns: sentience could organoid become conscious or experience suffering, consent or donor fully informed about how their cells will be used, experimentation limit what type of tasks are ethically permissible. Ethical framework must evolve to address these concerns. These includes monitoring complexity, watching for signs of advanced cognition or awareness, establishing safeguards, limiting experiment that could cause harm or cross moral boundaries, engaging ethicists involving experts in bioethics, law, and philosophy in research planning. Organizations like the Changer Project are working to create legal ethical guidelines for OI, but global consensus is still developing. Public perception. AI is a novel and complex field that may be misunderstood or fueled by the public misconception about brains in a dish or a living computer could lead to resistance or backlash. Public concerns include privacy. Could not derive from personal process or will sensitive information control? Who decide how voice systems are used and for what purposes? Equity. Will access to OI technologies be fair and inclusive? To address these concerns, researchers must communicate transparently, explain what organoids are, how they work, and what safeguard exists. Educate the public. Use outreach, media, and education to build understanding and trust. Include diverse voices, engage communities, patients, and stakeholders in shaping OI policy. Public perception will play a critical role in determining how OI is adopted, regulated, and integrated into society. Conclusion. Organoid intelligence holds immense promise, but it also faces significant challenges. Scalability and reproducibility limit its technical growth, data interpretation and control complicate its functionality. Ethical boundaries and public perception shape its societal impact. To overcome these hurdles, researchers must innovate in bioengineering and data science, collaborate across disciplines, engage ethically and transparently with the public. By addressing these challenges head on, we can unlock the full potential for ignite intelligence while ensuring it serves humanity responsibly and wisely. Future directions in ordinary intelligence still in its infancy, but its trajectory point toward revolutionary advances in biocomputing, neuroscience, and artificial intelligence. As researchers refine the science and technology behind brain organized, several key areas and emerging as priorities are for future development. These include increasing organoid complexity, creating standardized platforms for experimentation, integrating machine learning for hybrid intelligence, and building global network for open ethical research. Larger, more complex organoids. Current brain organoids are limited in size and complexity. Most are a few millimeters in diameter and contain hundreds of thousands to a few million neurons, far fewer than the 86 billion neurons in the human brain. To perform more sophisticated tasks, organoids must become larger, more structured, and more interconnected. Challenges to scaling: lack of vascularization. Without blood vessels, organoids cannot grow beyond a certain size due to limited nutrient and oxygen diffusion. Structural limitation: organoids often lack the full architecture of brain, such as distinct regions and long-range connections. Functional maturity: many organoids resemble fetal brain tissue and do not reach adult-like functionality. Solutions in development: bioengineered vascular system. Researchers are experimenting with synthetic scaffolds and co-culturing with endothelial cells to stimulate blood flow. Multi-organoid network connecting different organoids to stimulate inter-regional communication, long-term maturation protocols, and extending culture time and optimizing conditions to promote advanced neural development. Larger, more complex organoids could enable OI system to perform tasks like natural language processing, decision making, and even rudimentary emotional responses. They also offer deeper insight into brain development and disease. Standardized OI platforms. One of the biggest barriers to progress in OI is the lack of standardized platforms. Currently, different labs use varied protocols, tools, and analysis methods, making it difficult to compare results or scale experiments. Why standardization matter? Reproducibility ensures that findings can be validated across institutions. Scalability facilitates mass production and deployment of OI systems. Interoperability allows integration with digital tools, sensors, and machine learning model component of standardized platform organoid generation protocol uniform method for deriving and culturing brain organoid interface hardware multi-electrode array microfluidics and imaging system for stimulation and recording. Software tool, machine learning algorithm for data analysis, pattern recognition and feedback control, ethical guideline framework for consent usage and monitoring of organoid complexity integration with machine learning. Machine learning is a natural partner for organoid intelligence, while organoid offer adaptability and biological realism. ML provides speed, precision, and scalability together. They can form hybrid systems that learn, evolve, and perform complex tasks. Benefits of integration: enhanced learning. ML algorithms can interpret organized responses and adjust stimuli to optimize performance. Real-time feedback. Closed-loop system allow organizers to learn from experience, similar to reinforcement learning. Data analysis. ML tools can detect pattern neural activity, classify behavior, and predict outcomes. Use cases. Game playing as demonstrated in the Pong experiment, ML can translate game environment, stimuli and interpret organized responses. Medical diagnostic, ML can organize, revolutionize to drug identifying effectiveness treatments, foster cognitive modeling, hybrid system can stimulate aspects of human thoughts, emotions, and decision making. Researchers are also exploring neuro symbolic system where organoid handle in reasoning and ML manages symbolic logic. This could lead to AI systems that combine human life with machine precision. Global collaboration and open science. OI is a multidisciplinary field that spans neuroscience, bioengineering, computer science, ethics, and law to advance responsibly and inclusively. Global collaboration and open science essential. Importance of collaboration. Diverse of expertise, combining knowledge from different fields accelerates innovation, ethical oversight, international dialogue help establish shared values and safeguard resource sharing, open platforms and data repositories, democratize access to tools and finance. Key initiatives, Children Project, a European effort to create legal and ethical framework for OI research, open access journal publication like APL Open and Frontier Insight promote transparency accessibility. Global conference event bring together researchers, ethicists, and policymakers to shape the future of OI. Open science principle, transparency, sharing method, data, and result openly accessibility, making tools and knowledge available to all regardless of geography or funding, and inclusivity, engaging underrepresented communities and ensuring equitable participation. Conclusion. Why organized intelligence matters for the future of intelligence? Organized intelligence is not just a scientific novelty, it's a paradigm shift. It challenges our assumptions about what intelligence is, how it can be built, where it can reside. Reason OI is transformative, biological Realism, unlike digital AI voice systems, learn and adopt through natural neural process, offering insights into human cognition, energy efficiency. Biological neuron consume for less energy than silicon chip, making AI a sustainable alternative for future computing. Medical breakthroughs, patient-specific organoid enable personalized disease modeling and drug testing, accelerating treatment discovery. Ethical AI development studying organoids help us build AI systems that are not only intelligent but also ethically grounded and human-aware. New forms of intelligence AI open the door to hybrid systems that combine biological adaptability with digital precision, potentially surpassing current AI capabilities. In short, Organoid intelligence represents a new frontier not just in science and technology, but in our understanding of mind and machine. Organoid intelligence summary of key points. Organoid intelligence uses these organoids for biological computational learning. The field emerged after the discovery of induced pluripotent stem cells by 2006, enabling a personalized and ethical organoid computation. Background concepts. Brain organs mimic early brain development, exhibit neural activity, creating a reprogramming stem cell neural generator 2D culture compared to traditional AI/OI offers biological adaptability, energy efficiency, human life, host synaptic plasticity, uh, ethical and future direction, larger, more complex, organized, enhanced structures and connectivity, standardized platform, improve reproducibility and scalability, machine learning integration, hybrid system for advanced global collaboration, open science and ethical framework, cross-border conclusion, OI emerges biology and computing to redefine intelligence. It offers breakthrough in medicinal AI ethics. Call to action researchers, students, and policymakers must engage thoughtfully to shape its future. And thank you so much.